Every single second, millions of cells in your body are dying. It's happening right now. It sounds kind of grim, but it's a totally fundamental part of life. So today, we're going to investigate what it really means for a cell to meet its end. Because it turns out, not all cell death is created equal. Far from it. There are two main ways a cell can go, and honestly, they tell two completely different stories about what's happening inside our bodies. So on one side, you've got necrosis, and on the other, apoptosis. I want you to think of them as two very different case files we're about to open. Each one has its own set of clues, its own crime scene, and its own consequences. Okay, so first up is necrosis. This is the violent, messy end. Just imagine a building suddenly collapsing because of some catastrophic damage. It is totally uncontrolled, it's chaotic, and it leaves a huge mess in the neighborhood. But then you have apoptosis, which is the complete opposite. This isn't an accident. This is a planned demolition. It's quiet, it's incredibly orderly, and every single piece is carefully taken apart and packaged up for removal, leaving the site perfectly clean. All right, let's dive into the details, because this is where it gets really interesting. This table gives us our first major set of clues. Just look at the differences. With necrosis, the cell swells up and literally bursts, but with apoptosis, it actually shrinks down. The outer wall, the plasma membrane, gets completely destroyed in necrosis, but stays perfectly intact in apoptosis. And maybe the biggest clue of all? Necrosis is always a sign of something bad, disease, or injury. Apoptosis, on the other hand, is often a perfectly normal, even healthy process. Okay, so to really understand the story of necrosis, we need to zoom in. We need to look for evidence right at the very heart of the cell, the nucleus. This is where the most dramatic changes happen. And believe me, they follow a very specific pattern. And this diagram here, this lays out the entire chain of events for us. You see, a healthy nucleus goes through three distinct stages of breakdown. First, pycnosis, then karyorrhexis, and finally karyolysis. So let's examine each piece of this evidence, one by one. Our first clue is something called pycnosis. This is when the nucleus, the cell's command center, starts to shrink. All the DNA inside gets packed down super tight into this dense, dark little ball. It's really the first visual sign that irreversible damage has happened. And this slide is great because it connects the concept to what it looks like in reality. So on the left, you've got the diagram of that condensed nucleus. Now, with that image in your head, look over at the actual tissue sample on the right. See those super dense, dark dots labeled N? Yep, those are real pycnotic nuclei, caught right in the act. All right, let's move to the next stage of our investigation, karyorrhexis. Things get even more dramatic here. After shrinking down, that nucleus doesn't just sit there, it shatters. That condensed ball of DNA literally breaks up into a bunch of fragments. And again, we can see this happening for real. The diagram on the left shows the nucleus just shattering into pieces. Now look at the micrograph on the right. See those scattered dark fragments labeled K? That's the evidence. Those are the remnants of a nucleus right in the middle of karyorrhexis. And the final step in this whole sequence is karyolysis. Basically, those nuclear fragments get broken down by enzymes until they completely dissolve. The evidence of the nucleus just fades away. So on the left, the diagram shows what looks like a cell ghost. Its nucleus is totally gone. And if you look closely at the tissue sample on the right, you can see these faint pink cells that have lost that distinct dark nucleus. They've completed the process. They're done. So here's the crucial point. When pathologists are looking at damaged tissue, they don't just see one stage at a time. They see the entire crime scene. In a single view, just like this one, you can find cells with those shrunken pycnotic nuclei, others with fragmented nuclei, and some that are just those empty ghosts. It's a snapshot of total cellular chaos in progress. So now that we've followed the clues inside the nucleus, let's zoom back out. What kind of impact do these two types of cell death have on the surrounding tissue, on the whole cellular neighborhood? Well, the scene of necrosis is pure chaos. The cell's outer wall just ruptures. Everything inside, including these really powerful digestive enzymes from its lysosomes, spills out all over the place. And that leakage is a major alarm signal for the body. But the scene of apoptosis, it's calm, it's controlled. The cell membrane never breaks. Instead, the cell's contents are neatly packaged up into these little sealed bags called apoptotic bodies. There's no spilling, no mess, no drama. And this, Right here, this leads to the single most important difference in the aftermath. That messy, chaotic spill from necrosis triggers an immune response. It causes inflammation, swelling, redness, pain. But the tidy, contained process of apoptosis, it happens silently, without alerting the immune system at all. No inflammation, none.
So, we've examined the evidence, we've seen the aftermath. What's the final verdict in our investigation? Why is it so incredibly important for us to understand the difference between these two ways a cell can die?
This quote from our source material just nails it. Necrosis is always a sign that something has gone wrong. It's the result of an injury, or a poison, or a disease. It's bad news. Apoptosis, on the other hand, is often a good thing. It's the body's way of sculpting tissues, getting rid of old cells, or eliminating cells that could turn cancerous. It's absolutely essential for a healthy life. And that leaves us with this final, really fascinating question to think about. If our bodies absolutely depend on this orderly, planned process of cells suicide just to stay healthy, what happens when it breaks? What are the consequences when the cells that are supposed to die don't?